Hi, my name is Rebecca Reed, and I'm the Outreach Manager at the Fort Worth Museum of Science and History. I'm so glad you could join us today. Did you know that you can extract DNA using simple things you have at home? Well, you can. Today, we're going to use strawberries, but you could also use wheat germ or bananas. Before we get started, let's talk a little bit about what DNA is. DNA stands for Deoxyribonucleic acid. It's a molecule that carries the genetic instructions used in growth, development, functioning, and reproduction for all known living organisms and many viruses. DNA and RNA are nucleic acids, and alongside proteins, lipids, and complex carbohydrates, they are one of four major types of macromolecules that are essential for all known life. Most DNA molecules consist of two strands coiled around each other to form a double helix. The two DNA strands are called polynucleotides, and they're composed of simpler units called nucleotides, and there's 4 different kinds of those. They're joined in a chain of covalent bonds between the sugar on one nucleotide and the phosphate on the next. It results in an alternating sugar-phosphate backbone. The nitrogenous base of the two separate polynucleotide strands are bound together according to the base pairing rules. And they use hydrogen bonds to bind together. And this makes the double-stranded DNA. DNA is inherited by children from their parents. This is why children share traits with their biological parents, such as skin and hair color and eye color. The DNA in a person is a combination of the DNA from each of their biological parents. Now that we know a little bit more about DNA, we can begin to extract it from our strawberries. Strawberries are particularly good for this lab because they're soft and they're easier to squish and pulverize, and they yield a lot more DNA than other fruits due to some genetic traits of the strawberry. Now we're going to start extracting our strawberry DNA. You're going to need 5 milliliters of rubbing alcohol, 10 milliliters of dish soap, a 1/4 teaspoon of salt, 90 milliliters of water, and some strawberries. You're also going to need some equipment. You're going to need a zipper bag, a strainer, some measuring tools, glass containers, and some toothpicks. Now to get started, the first thing you're going to do is you are going to take one of your glass containers and you're going to add first your 90 milliliters of water to that. I had to put that into 2 different containers because mine's a little... You want to make sure that your container's large enough to hold it. So then, next thing you're going to do is you're going to add your dish soap. That's a little tricky to get out, so just do your best. You may need to hold it a little bit. You may want to use something to scrape it out. And I have a stirring stick I can use. So we're just going to stick that in there and kind of scoop out all that extra soap. There we go. And then the final ingredient we need to add to this is our salt. So you need to add 1/4 teaspoon of salt. Just regular table salt, nothing fancy. You can add that. Then you want to stir these until they dissolve. Now, you don't want to stir it so much that you get lots and lots of foam, but you do need to stir it until all of that salt is dissolved in the liquid. And it'll take just a little bit. You can see mine's already starting to get all dissolved. Okay, you've made your extraction liquid. Now the next step is to take your strawberries and to put them into a plastic zipper top bag. And you'll need between 1 and 3 depending on the size of your berries. Overripe berries or really squishy berries work the best because they're the easiest to mash up. Then, once you get your berries in the bag, you're going to add your extraction liquid. So you're going to want to make sure that your bag doesn't have any holes before you do that. You're going to zip it shut. And then you're going to start squishing. And you want to try to get all of the chunks out. And you're just going to keep squishing and squishing and squishing. Once you've squished up all your berries, you should notice your extraction liquid has turned a pink color, just like your strawberries. You're going to want to set that aside and get your strainer and a glass container ready. And you're going to pour this strawberry mixture right into that container. And all the strawberry pulp too. Everything's going to go. Once you've got that all situated and all poured out, you're going to need to force the rest of it through the strainer. And so you may want to use a spoon to help do that, or you can just use your fingers. But you want to try to get as much of that liquid, as much of that strawberry mixture down into your glass container. Once you've got most of that through, then you're going to move on to the last step. And you're going to be taking your strawberry mixture and you're going to pour it into a smaller glass container. And you want to put about 1/2 cup to 1/4 cup in there. So that's about anywhere from 80 to like 110 milliliters if you're using metric. So you'll pour that in. And then the last step today is you're going to take some ice cold rubbing alcohol and you're going to pour that on. And then you have to wait and see. You should see some separation. And what you'll see is a kind of a clear white layer start forming at the top. Then you can use a toothpick if you want to kind of scoop that up. And this stringy layer right here, that is actually the DNA from those strawberries. Congratulations! You've extracted DNA. You'll notice that it's not in those long strands we talk about. You kind of see it as little clumps, and that's just part of this extraction process. I hope you enjoyed this lab activity. If you have any questions or other ideas for videos, please email us at questions@fwmsh.org. Thanks, and we'll see you next time.